So till now we have discussed about the different terminologies, the diagrams, the processes. Okay, and we are almost towards the end of the chapter. And I've told you again and again, go through the NCRT once again. And all the words, lines, terminologies are quite very important because CBSE expects you to answer the questions as per the NCRT. Okay. So in my last class, I was discussing about the double fertilization process, and I talked to you about this is the diagram that is related to the this is the diagram that I'm talking about. It's fertilized embryo sac we talk about. Okay. And this fertilized embryo sac is the diagram is a bit different from the mature embryo sac. So make sure that you are reading the question also very carefully and you know that exactly what needs to be given. Okay, so when I'm talking about the fertilized embryo sac, I'm talking about two very important things. Okay, now before that, this is the micropylar end towards which I'm writing M for it, and this is the callosal end. On the micropylar end, you have a zygote present, which is represented by the 2n number, and rest of the cell structure, that is the synergids, these are getting degenerated. You can show it through a membrane that is getting degraded. Okay. And on the other hand, you have antipodal cells towards the callosal end that is also getting degenerated. In between, you have endosperm cells. So yesterday I was telling about the double fertilization. What does that mean? There are two types of fertilization that is happening. One is the male gamete fusing with the egg cell that is forming what is known as a zygote. And second one is the second male gamete fusing with a polar nucleus. So it is already 2n in number, so together it becomes 3n. So this is the diagram of a fertilized embryo sac and how you need to understand it also. Now there are two terminologies are being used. Okay, one is known as PEC. One is PEC, and this PEC is a primary endosperm cell. So sometimes the question comes, what is the haploidy of the PEC? That is what is the ploidy of the PEC? It is 3n in number, right? Make sure about this thing. And what is the PEC finally developing into? PEC is finally developing into an endosperm. Remember this thing. The PEC, what is the fate of PEC? The PEC develops into an endosperm. What is the endosperm doing? What is the endosperm doing? It is providing nutrition to that growing embryo. What is happening to the other structure? That is a zygote we talk about. What is the ploidy of the zygote? It is 2n in number. So what do we say this as a zygote? This is, right? And it is 2n in number. What is the fate of the zygote? The fate of the zygote is that it is getting converted into an embryo, right? So very small questions are asked regarding this thing, the fate, the ploidy, and what happens to these things. So we are talking about the fertilization. That is how we talk about the double fertilization. What is a product of fertilization? It is endosperm. What is the product of the second fertilization? Is the embryo, means the zygote formation, which later on forms the embryo. Okay, fine. So this was all about the fertilization event. We next come to what happens post-fertilization. Okay, so once the fertilization is done, what happens to the endosperm? Obviously, in this case, we talk about two, three different things we talk about. One is the zygote getting converted to the embryo. So when you talk about the zygote getting converted to the embryo, it's a bit different both in case of a, you can say a dicot plant and a monocot plant. There are two diagrams in front of you. I'll explain you both the diagrams. The first diagram is of a dicot embryo, right? So how do you know it's a dicot embryo? The dicot embryo has got two cotyledons. Can you see over here? The labeling part, it has got 1 and 2. So these are the two cotyledons present in the dicot plant. Okay. And the cotyledons are then having the plumule region. What is this plumule region? This plumule region, when the seed germinates, it becomes a stem part. Okay. And the other part is known as the radical part. This radical part is something which will become the root. And above the radical part, we have a covering which is known as a root cap. That is known as, okay. The first diagram is showing you the dicot embryo. It is showing you, okay. And what is that embryo? Actually, it is a division through which the zygote undergoes. And this division creates zygote, becomes a multicellular structure. And then we start calling it as an embryo, okay. And that process is known as embryogeny. That process is known as, okay. So the initially, if I'm talking about, let us just once again go back to the one diagram. What is that embryogeny? It is the formation of the embryo we are talking about, okay? So make sure that the formation of the embryo in the initial stages, it is similar in both dicot and the monocot plants, okay? So you can see over here, there are different structures given to you. Again, this whole process. Can you see this is a zygote? This one cell structure having one nucleus is zygote. Beyond all these structures, they will be now known as what? They will be known as embryo, right? And the process of formation of the embryo is known as embryogeny. This is known as. So the initial stages in both the zygote and the monocot, it is similar, and that is called the embryogeny. That is known as, you have different type of embryos are there. You have a globular embryo is there. There's a heart-shaped embryo is there, and finally, okay. And once the stage is passed, okay, once the stage is passed, it gives rise to a different pattern of the embryos, you can say, and it is a bit different again in the case of a monocot and a dicot. Okay, so when we talk about the monocot and the dicot, you have both the regions. You have the plumule and the radicals are there, and that is how later on, when the seed germinates, it forms the stem and the root part. Now, very important is a diagram of a monocot embryo also, right? They've given you the structure of a LS of a grass embryo. Actually, they have given you, okay, a very microscopic small structure it is. They have given you the lateral section. LS means the longitudinal section, sorry, the longitudinal section that is. And this longitudinal section of the monocot, it generally comes as a 3-marker question or something like that. It comes in examination. So make sure you're thorough with this diagram also, okay? I'll explain you one by one. So what is the meaning of the monocot? It means that it has got just one cotyledon in it, right? Remember one point. What is a monocot? It has got just one cotyledon in it. The monocot's cotyledon has been given a new name, and that is called scutellum, that is known as. Remember this thing. Once again, I'm repeating what I'm talking about. We are talking about the embryo of the monocot, okay? If somebody is asking you, is the embryo of the monocot and dicot very different? We say yes. In the initial stages, when you talk about the embryogeny, in the initial stages, when the heart-shaped structure is there, globular structure is there, they're all similar. Initial stages of both the dicot and the monocot are similar. Later on, it become a bit different, okay? The cotyledons are 2 in number in the case of the zygote, in the dicots, and it is 1 in number in the case of the monocot. And remember that in case of a monocot, okay, we say that the cotyledon has been given a new name over there, and that is called the scutellum, that is known as, okay. So this is important. Scutellum is an important word. And what is the scutellum? It is the cotyledon of the monocot embryo, okay. Apart from scutellum, you can look at the diagram. This is of a grass family they have given, okay. And this grass family has got 2 different more other structures are there. We'll talk about these structures, right? Okay. So these structures are, number 1 is this region. If you can see, this is the shoot apex part, right? It is going to later on become the shoot part. So we call it as a shoot apex. And this whole of the shoot apex is surrounded by a structure that is called the coleoptile. Right, now, what was the scutellum? It was a cotyledon, and that is having an embryonical axis that is known as on the embryonical axis. That is, apart from that, you have a shoot apex region. Above the shoot apex, the layer which is covering the shoot apex is known as a coleoptile. That is known as, okay. On the other hand, towards the lower side, you have another region that is called the radicle. That is going to be the future root, also with the root cap region. So radicle and the root cap region is towards the lower end. So if you can see, this is a radical part, right? Where I'm just doing it like that, the orange color, you can see this is a radical region, okay? And below this radical region, this region is actually the covering of the radical that is called the root cap, that is known as. The way shoot apex was covered with the coleoptile. In this case, the root cap or the complete radical, it is covered by structure that is called the coleorhiza, that is known as, okay? So when you are labeling, make sure that all the labels are properly placed, okay? And one more structure is there present, which slight depression is there, and that depression in between the coleorhizas and the coleoptile is known as epiblast part, okay? So scutellum, then we have a shoot apex. The shoot apex is surrounded by the coleoptile. Then we have the radical. The radical is surrounded by the coleorhizas, okay? And what is the epiblast? In between these coleoptiles and the coleorhiza, there is a depression-like structure present in the embryo when we cut the longitudinal section. And that longitudinal section being cut and that depression is known as an epiblast, that is known as. Fine. Okay, let me proceed from here then. Let me proceed from here. Okay, fine. So I think now it is clear. Sometimes the question comes where you have to compare between a monocot and a dicot embryo. So I think 2-3 terminologies can be very easily explained. Okay, number 1 is that the dicot has got the 2 cotyledons in it. Okay, Then you have one more, this thing is that the cotyledon of the monocot is given a new name, that is a scutellum. And then the presence of the coleorhizas in the coleoptile. What is the coleoptile? Surrounding the shoot apex. What is the coleorhiza? It is surrounding the root apex. Okay? So we have this as a embryo discussion over the embryo. Once the zygote formation happens, it divides and it forms the embryos. Okay, fine. So all the diagram need to be practiced. And see, it's not like that you are just looking at the diagram and you'll be able to draw it in the examination. Make sure you are practicing the diagram, okay? In a rough copy, keep on practicing it so that you get a diagrams in examination should never be made like a rough diagrams, okay? Being a biology student, you all should be drawing a very proper diagram so that it's not like that the marks are going to be cut or something like that. Not sure about it. Sometimes the rough diagrams have also been given marks, but according to me, my suggestion would be to always give a very proper and a well-defined diagram. Okay. So whatever is happening till now, the embryo formation has happened, the fertilization has happened. We discussed about the all the pre-fertilization, even we discussed about the fertilization. Even now, the major thing is now happening, and the sexual reproduction is going to end over here. Okay, it ends in the case of the angiosperm. The reproduction process ends with the formation of the seed. Remember, right? This can also become one marker question, that what is the final product of a sexual reproduction in case of an angiosperm? So actually it is the seed formation, remember, right? So when the seed is being formed, we said, yes, the things are now getting completed. What is actually there where the seed is present? Seed is inside the fruit. In common example, you can say, take it of a mango actually. In mango, you can see the part which you eat actually, that's a fruit. And if that part is completely, you've eaten all the complete fruit part, what is there inside? It's a seed inside, okay? So where is the seed present? It is present inside the fruit, okay? What is seed? What is a seed? Seed is a, one more word is term or explanation part can come over here. Seed is a fertilized ovule, that is, right? Seed is a fertilized ovule, okay? And what is a fruit? Again, when we talk about the fruit, what is a fruit? Fruit is a mature ovary, right? So again, I'm talking about the fate of the ovary, the fate of the ovule. So the ovary becomes a fruit. What is a mature ovary? It's nothing but a fruit. What is a fertilized ovule? It is a seed. Now, I think this is clear. So make sure that you're not mixing up your words. Okay, you know exactly know that how where the seed has originated from. It has come from the ovule part. Okay, where the fruit has come from, it has come from the ovary part. So mature ovary is a fruit, and a fertilized ovule is a seed. It is okay. Fine. Now, next thing when we talk about is the what does this seed actually contains, and what are the different type of seeds we talk about. So we have a non-albuminous and or ex-albuminous seeds are also present. Okay, like for example, what I said is that whenever a zygote formation is happening, before the zygote formation, there's an endosperm formation happening. Okay, this endosperm is providing nutrition to the growing zygote. Because as the zygote is getting converted with embryo, it needs to be given nutrition. A non-stop nutritional supply needs to be given. So this endosperm is providing nutrition to the growing zygote, to the growing embryo. Okay. Finally, when the mature seeds comes up, okay, maybe it is not having any residual endosperm with it. Okay, such type of seeds, if it doesn't have any residual endosperm with it, are known as a non-albuminous or an ex-albuminous seed. Right? I'm repeating once again. What are we talking about? When the zygote formation happened, at the same time, the endosperm formation also took place, right? And the endosperm was used by the growing zygote, by the growing embryo in the form of a nutrition. So when the mature seeds are developed, they are actually without that endosperm. There's no residual endosperm left, okay? Because it is completely used up by the developing embryo, as in the case of a peanut and the groundnuts they are talking about, okay? Such type of seeds are being classified as a non-albuminous or an ex-albuminous seed. I think this is clear, okay? On the other hand, if I'm talking about the examples of the wheat, maize, barley, castor, the four examples that book has given, okay? Wheat, maize, barley, and castor. What's happening in these seeds, okay? So as in when the zygote is getting converted to the embryo, okay, the complete endosperm is not used, right? So there is some part of the endosperm which is still there in a mature seed, okay? In this particular type of seed, are actually known as the albuminous seeds. These are known as. So on one side, you are having the one seeds like peas and the groundnut that don't have any endosperm with it. On the other hand, you are having the seeds like wheat, maize, barley, castor, which are actually having endosperms. So that is how we classify the albuminous and the non-albuminous seed. Okay. There is one more terminology. I'll just stop sharing this. I'll open my whiteboard. One more terminology is being used when we talk about such type of seeds we talk about. Okay. So I talked about number one, it was an albuminous seed that was albuminous seeds. Okay. So, and second was ex-albuminous seed or a non-albuminous seed, albuminous, right? One more terminology is there, and that is called the perisperm. I would like to discuss all three together. So now you've understood, like if I'm talking about the ex-albuminous, we talked about the pea, and a groundnut, we talk about albuminous, we talk about the wheat, na? We talk about maize. All these were actually having endosperm within them. Okay. The last one is the perisperm. Now, what is this perisperm actually? Okay. If a seed is not only having an endosperm, it is also having a ruminant of a nucellus. If you'll remember, nucleus was surrounding the new embryo sac region. It is made up of the parenchymatous cells, okay? And it is surrounding the embryo sac region. If a mature seed, remember this thing, we are talking about a mature seed now. In all three cases, what are we talking about? We are talking about a mature seed. So mature seed with the endosperm is known as albuminous. Mature seed without endosperm is known as ex-albuminous. Okay, and a mature seed with a reminiscent nucellus also. What was the nucellus? It is the embryo sac. If you remember the mature embryo sac, the diagram which we did, this was the integument part, okay? And around the integument part, the inner layer, this was the nucellus layer. Again, surrounding the embryo sac, providing nutrition to it. If a mature seed is still having, it is not being consumed totally, it is still having the nucellus inside it, then it is known as a perisperm. It is known as, right? Now, if the example of the perisperm is a black pepper, if you have seen this black pepper somewhere, just look at the seed of the black pepper, right? It is slightly, you can see the crevices on the surface of the black pepper. That's the nucellus part we see, okay? So what is that black pepper? It is having the remnant of the nucellus inside it. And we give one important terminology to it. We say that it's a perisperm structure that is present. So you have albuminous, you have the ex-albuminous, and we have the perisperm structure is there. All the three structures are present when we talk about the mature seed we are talking about. Okay, fine. I'll come back to my next sharing. So I think the three terminologies are very clear in your mind. We talked about the non-albuminous seed, we talked about the albuminous seed, and we talked about the this thing, the perisperm. Okay, now what other things are present over the ovules or the mature ovules or the seed? What are the things present? Okay, one more very important thing present is a seed coat we talk about. Right, we talk about a seed coat. We talk about what is the seed coat? It is the integuments, right? Integuments later on they become hard, and what they form, which is known as the integuments, are actually again questions come like this: what is the fate of the integument? Integuments actually form the seed coat. It actually becomes the seed coat. Hardened seed coat is present over there. Okay. Again, an important thing, what happens to the micropyle region? So it is present in the form of a very small pore in the seed coat. You just look at the seed of a chana or something like that. You will find a very small pore is present. From there, the water and the oxygen gets inside the seed during the germination process. Okay. That small pore is actually the micropylar region that remains open in the case of a seed coat also in the region of the seed coat, and becomes a very small opening. And this opening is helping in the germination process. Whenever you make a sprout, you know this thing, that when the things are getting germinated, what's happening actually? It is taking up the moisture, it is taking up water, it is taking up oxygen, and that is how the division process happens. That is how the seed gets germinated. So where this oxygen and water is coming inside, it is coming through this seed pore, you can say, or the opening, and which is this part representing? It is representing the micropylar region. Okay. Again, what happens is that when the seed is maturing, okay, when the seed is maturing, and there is not enough water around it, there's no place for it to actually germinate. Okay, slightly, slowly, what happens is that the seed becomes relatively dry, right? When it becomes dry, the inside activity of the embryo also slows down, okay? And the embryo is not dead actually. See, whenever we give it back its favorable conditions like oxygen and water, it germinates. So we won't say that the embryo is dead inside it. But what happens to the embryo inside it? It metabolically slows down, right? So what is happening is that when a seed is maturing, remember this very important thing which I'm talking. When a seed is slowly maturing, it is losing its water content, it's losing its moisture content. Okay, when this moisture content is losing, the seed is losing, it is actually drying up, right? This phase of the drying up of the seed is actually making the embryo metabolically inactive in nature, and that is the condition which is termed as dormancy. Very, very important. This allows the seed to actually cross that unfavorable condition where the water and the availability of the oxygen is not there. Fine. So what happens to the embryo inside the seed? The seed has got hardened up. Okay. The seed has got dried. It has lots of moisture content, but the embryo inside it doesn't die. Okay. It actually enters into a state which is where it is metabolically very less active. Almost it's like an inactive structure it is, and we say that the seed has become dormant in nature. Okay. And once again, after later on, sometimes you provide like the way we make sprouts at our home. Okay, you provide water, you provide optimum temperature, you provide oxygen to the seed. Once again, it starts germinating, right? And what is the germination process? Once again, it starts dividing, the cell starts dividing, so it breaks its dormancy once the favorable conditions are attained. Okay, so I think this is very clear. Every term, I think, say, the terminology can become a question. What is the seed pore? Where does the seed pore comes from? It comes from the micropyle. Okay. What is the seed coat? It comes from the integument. Okay. What is this dormancy process? Why the dormancy process attained? Okay, because the seed is losing its moisture content. It is drying up as it is growing, developing. It is drying up. So the embryo activity stops inside it, and this inactive embryo is now under a dormant condition. Once again, you give back oxygen and temperature to it, it once again starts dominating, means it once again start dividing inside. Okay. Apart from the ovule, okay, what we have in the inside around the seed, we have the fruit. So what is that fruit? Fruit is a ovary, right? The mature ovary is a fruit. What is the wall of the ovary? What is the wall of the ovary? That develops into that is called the pericarp. Remember this. So, like for example, they have given examples of guava, orange, mango. The peel which you take outside the fruit, that is actually a pericarp, right? So wall of the ovary is becoming nothing but it's becoming converting itself into a pericarp. It's a covering. And the peel you take out of the guava, or you see outside the orange, or you see inside outside the mango. Okay, what is all those things? This is a pericarp, that is okay. It's the same. Or maybe in the dry structures also, like groundnut, mustard, also you take out that peel. Okay, what is all these structures? These are all the pericarp, which is actually the wall of the ovary that's got converted into the pericarp region. Okay, fine. So many questions do come in examination, which talk about what is the fate of the structures, right? They talk about what is the fate of the structure, what happens to these structures, and what happens, where does the seed coat comes from, what happens to the integument, where the pericarp is made from. So make sure you are able to relate everything, right? You're able to relate. What is this pericarp? It's a wall of the ovary. What is that fruit? It is a mature ovary. What is that seed? It's a fertilized ovule. So all these answers should be there in your mind. Okay, fine. Next we have, uh, okay. So when we talk about the fruit, okay, in terms of biology as a subject, okay, we need to decide what a fruit is actually. Basically, when we say a fruit, okay, we say talk about the true fruit we talk about. The true fruit should have been developed from a ovary part only. So means mature ovary is getting converted into a fruit. Under that condition, we say that it's a true fruit that is. Okay. See, what are we saying is that, but like for example, the cases like cashews, strawberries, and the apple, they have said, okay. Just a minute. Let me open a white screen. Right. So this thing needs to be very clear in your mind that when I'm talking about a fruit, I'm talking about, okay. So What is the origin of the fruit? So fruit originates from a mature ovary. Right? Now this is very clear in your mind that where is the fruit coming from? It is coming from a mature ovary. Right? Now the fruit is further dividing into two types. Fine. On one side, we talk about a true fruit. On other side, we talk about a false fruit. True and a false fruit की हम बात करते हैं. What is a true fruit? If the fruit is developed totally from the ovary part, right? Like for example, guava, they talked about. I think they gave an example of a mango, guava. Okay. These are all the examples of a true fruit because that fruit is coming from a mature ovary. I'll use the word mature ovary over here. Right? The ovule has got fertilized, the ovary has got mature, so now it is forming the fruit. Okay. What's happening in the case of a false fruit? They've given an example of an apple. Now, apple is not a true fruit. Okay. In terms of a biology, it's a false fruit. Then we say cashew is there. So all these structures on apple and cashew, they no doubt the ovary is becoming the fruit, but along with the ovary, the fruit is also having part of a thalamus region, right? So in this particular case, we can say that the fruit is just not being made from an ovary. Something else is also being added to it, and that is the thalamus part is being added to it. Such type of fruits, as in the case of the apple, as in the case of the cashews, okay, such type of fruits are known as a false fruits. Example, where we talk about is in which the ovary and the thalamus both are included and both are becoming the part of a fruit. We say that such type of fruits are not the true fruit, they are the false fruit. Okay. So I'll go once again. What is a mature ovary? Mature ovary is a fruit. The fruits are divided into two different parts. We have a true fruit and a false fruit is there. What is a true fruit? It is the fruit that is just made only from the ovary, right? The mature ovary getting converted to totally to the fruit, that becomes a true fruit. Example, guava and the mango. On the other hand, we are having also the concept of a false fruit, like in the case of the apple and the cashew. What's happening in it? In this particular case, it is not only the ovary, but along with the ovary, one more part of the flower, that is the thalamus, okay? That also becomes the part of the fruit, okay? And that is how we say that it is a false fruit, that is. Fine? Okay. I think this concept is very clear. Again, I'm telling you, not very big and large questions come from this chapter. From these topics, especially the topics which are now we are ending on to it, okay? But obviously, every small little bit of concept that is present over there, you need to understand it very properly. Okay. Now, let us come to one more concept. Before coming to the another concept, there are certain diagrams given. Generally, CBSE doesn't ask for these diagrams, okay? But maybe they are just talking about different types of seeds they are talking about, right? So different type of seeds are like, for example, in the case of the first case, they talk about the monocot seeds are there. You have the dicot seeds are there again. Different types of seeds they're talking about where you find the cotyledons, the seed coats, the endosperm, the root tip, the hypocotyl, all these things. In this case, hypocotyl, the shoot apical meristem, root tip. So these are all different type of seeds they are talking about. Okay. Along with that, there is one more concept when we talk about. I think they've given you example of a false fruit. Yeah. Again, they're talking about the false fruit also, where inside is a seed. Now they have opened the apple and shown you. Okay. Now, in this case of an apple and a strawberry, what the concept is that this inner structure is the seed. Can you see this whole structure of the seed? Now, the seed is not completely surrounded by a mature ovary. It is also having a thalamus part also, right? So you have the ovary. यहीं पर खत्म हो गया पूरा ovary वाला part. You have the endocarp, mesocarp, pericarp. This whole part is got complete. What else is there? Outer part is covered with the thalamus region. That's why we say that apple is a false fruit because of the presence of this thalamus in the fruit. Right? This is a structure outside the ovary present, but it is still becoming the part of the fruit it is becoming. So we say that this is actually a thalamus part which we talk about. And this thalamus part is having the seeds inside it. So the fruit is not a true fruit, it's a false fruit. Again, in the same case, they've shown you example of strawberry also. You can see strawberry also they've shown you the structure where the inside part is actually not the ovary, but it is again a thalamus part. So in any of the fruit where there is this thalamus region is located, all such fruits are not a true fruit, but they are all the false fruit there, right? So I think question can come like that. They just give you the diagrams and they ask you to label it, and they ask you to tell you that whether it's a true or a false fruit. Okay. So obviously, whenever you are labeling thalamus in a fruit, okay, make sure that such type of fruits are not considered as a true fruit, but these are all the false fruit. Okay. Now, one more concept comes into picture. Okay. Let me once again. There's one more terminology along with the fruit I need to discuss. Okay, the one type of fruit you can talk about. Okay, we say that there's a concept of what is called parthenocarpic fruit. Parthenocarpic parthenocarpic fruit we are talking about. Now, this is very interesting, okay? We all know that when we talk about a fruit, we always talk about it as a post-fertilization event, okay? Formation of the fruit is not a pre-fertilization event. It's actually what's happening after the fertilization has happened. The ovule has got matured, the seed formation has happened, then ovary has got matured, and then we say that the fruit formation has occurred. You can understand these things happen after the fertilization process. Once the seed is being made, okay, then after the fruit is being developed. There are certain cases, like in the case of a banana. You must have seen when you open a banana, you'd hardly find any seed inside, right? So banana is an example of a parthenocarpic fruit where actually the fruit formation has happened, but without fertilization. Means there's no fertilization that has happened, but due to certain hormonal changes, what has happened is that the fruit formation has occurred in the banana, okay, but not as a part of a post-fertilization event, okay. It is just being made because of some hormonal change. The ovary got matured and it formed a fruit without the seed formation because no fertilization has happened. So there is no seed formation in it. Now, this is very interesting, right? And such type of fruits will be known as a parthenocarpic fruit. Such type of fruits are known as. In today's time, like for example, the seeds I talk about the watermelon. If I talk about any of the fruit which is having not many seeds inside, it's just a waste of time that instead of a person getting the fruit, he's just wasting his time removing the seeds actually. Okay. So for commercial purpose also these days, most of the like papaya, watermelon, okay, there are not many seeds inside it. Okay, what happens if there's no seed? What does that mean? Okay, so what are we doing? What are we doing is that we are creating a seedless fruit. We are creating. Okay, and how do we create a seedless fruit? There are certain hormonal changes that happens in such type of plants. Okay, and it creates an environment for the ovary to get matured, to get converted into the fruit without the seed getting formed. There's no fertilization that has happened. So you have a banana without a seed, you have a watermelon without a seed, you have papaya without a seed. Okay, how these things are being made? Because the concept is that you mature the ovary, right, without the seed formation, without the fertilization. And such type of formation of the fruit without the seed and without the fertilization is termed as a parthenocarpic fruit. Right? So remember this thing. This is again an important thing that we talk about. The terminology, like we talked about true fruit, like we talked about a false fruit. We also talk about a fruit that is called the parthenocarpic fruit, where there is no seed formation, right? Now, if you want to actually make a seedless, what do you say? If you want to make a seedless fruit, right? Like I talked about a watermelon, I talked about a papaya. If I want to make a seedless fruit, what I'll do is that I will be using the technique where the mature ovary is getting converted to the fruit without the fertilization process. Means there is no seed for because seed will be formed only after the fertilization, right? There is no fertilization that has happened, but the fruit has got matured, and such type of fruits are known as what? They are called the parthenocarpic fruits. These are known as. Okay. Fine. Let me just stop sharing this thing. So almost we are towards the end of the chapter, and this chapter actually holds a many weightage it holds. Okay, so among the few very important chapters it is, and that's why I took this chapter in the beginning and these all things. Okay, And apart from the parthenocarpic fruit, then they talk about what is the seed dormancy. They've talked about certain things they've talked about where germination of the seeds is happening. See, what was the last process? Okay, what was the last process? The seed will get favorable condition. Under this favorable condition, it is going to divide, right? It is going to germinate. And through the germination, it is going to produce radicals in the premiums. And obviously, you're going to get a new plant you're going to get, okay? So this is how we talk about. Now, this is very important that how does the seed get germinated and what's the time lapse, okay? So it's nature has preserved the seed for a very, very long period of time, okay? Many species of the seeds can actually live for hundreds of the years. They've given an example actually of the oldest of lupine, Lupinus arcticus, excavated from the Arctic tundra. Right now, you can understand that Arctic tundra is having a very low temperature over there. So the seed got protected, and was for like hundreds of the years it got protected. Okay, and this seed germinated and flowered also. Not only germinated, when it received the favorable condition, it germinated and flowered, and a record 10,000 years of dormancy. Right. So actually, the plants have this technique of preserving the seed because seed is their next generation. So till the time the plants attain that favorable condition, they're going to preserve that seed, and that preservation is done through a very, very interesting phenomena, and that is dormancy. Right. Remember this term, dormancy, and the dormancy can undergo for like 10,000 years they are talking about. So you can understand 2,000 years date from recently they've discussed the 2,000 years old viable seed. When we use the word viable seed means the seeds are actually alive and they can, given the favorable conditions, they'll actually germinate. Okay, such type of seeds are known as the viable seeds. These are known as, okay. Again, there is one more, Phoenix dactylifera. The seed of the date palm Phoenix dactyphylla was it was like a 2,000-year-old seed. This was excavated near the Dead Sea region. One was in the Arctic tundra region. That was a lupine. Another one is in the date palm of Phoenix dactylifera that was taken out in the Dead Sea region. So how these seeds are being preserved? They all are getting preserved through the process of the dormancy, where the embryo inside is not dead, right? It is not dead. It is metabolically inactive actually. And this metabolically inactive are embryos are actually, what are they doing is that they are inside the fruit, and later on when the favorable conditions comes up, they actually they germinate. Fine. Okay. Few last terminologies from this chapter. Okay. Okay. Like we said that the fruit is developed without the fertilization, we use the term parthenocarp. There is one more term being used where we use the term, one more term we use. And we say that apomixis. Now, what is that apomixis condition? Okay. When a seed is developing without a fertilization, right? Now you can understand what's happening. Like the development of seed happens once the fertilization happens, all these things are happening. But there are few species of the grasses and all those things that is, they keep on producing the seed even without the fertilization has happened. Okay. And this condition is known as the apomixis condition. These are known as, okay. So make sure that you remember this term also. Again, in apomixis, they've given an example of the Asteraceae family and the grasses. These are the family of the grasses and the Asteraceae. They do this process that they form the seed, but without the fertilization from. Formation of a fruit without a fertilization is a parthenocarpy, right? Formation of a seed without a fertilization is a apomixis process. One more concept there is of a polyembryony also. Again, NCERT is not mentioned much about it, but just the terminologies are there, so you should be knowing. 1 or 2 marker questions come from this topic, okay? So inside the embryo, for example, inside a seed, there is more than one embryo is present, okay? A lot many embryos are present inside the seed. So in this case, we use the term polyembryony from there, right? So there is one seed, but within that one seed, you have lot many embryos are there, more than one embryos are there. Then we use the word polyembryony, okay? So you can observe this in orange, that complete one seed, you open it, you get so many embryos inside it, okay? So that's a condition when we talk about is in. Always remember that you're always giving example. When you are explaining apomixis, give an example. When you're explaining polyembryonic, give an example. Parthenocarpy, give an example. albuminous seed, ex-albuminous seed, whatever you're talking about, okay? Make sure that every time you are discussing something, you are defining something, you are always giving an example. Obviously, once again, this is the end of the chapter, so make sure that you are going to the diagrams very carefully, diagrams of the labeling part, okay? Draw the diagram again and again. Don't just leave it like that, okay? And make sure that all the diagrams are being made. Explain it properly. Terminologies are very important. The chapter is not very, very lengthy chapter, but obviously, very, very small mistakes students are doing. Like I told you about the mature embryo sac, fertilized embryo sac. What is the difference between the two, right? So make sure that all these things are you are managing well. Okay. So that's how I end this chapter today. In my next lecture, I'm going to start with my another chapter, and that is going to be human reproduction. We'll talk about again a very, very important chapter, and the chapter that holds a lot of weightage. And obviously, I would say that it's a very scoring chapter. Okay. So in that case, thanks all of you for joining. Thank you. That's how I wind up my chapter. Thank you.